cholesterol synthesis. So, what is the significance of cholesterol synthesis? Cholesterol has many functions in our body. Okay, so it is a major lipid constituent in our body, apart from the phospholipids, because phospholipids make up the major component of the cell membrane. But cholesterol is also very much essential for the integrity of the cell membrane, right? So when we take food in the form of carbohydrates, they get broken down into glucose, and we have already discussed how the glucose will be utilized for production of pyruvate, lactate, acetyl-CoA, and ultimately entering the citric acid cycle to release energy. So not all the glucose will be utilized for energy then and there, right? So we would be eating food which may be above the demands of our body, above the energy demands of our body. So what the rest of the glucose and the acetyl-CoA produced, where it should go? So they would be converted to other compounds and get stored in our body, so that at times of need, we can reutilize them. Okay, so that is where cholesterol synthesis, fatty acid synthesis, ketone body synthesis, and all comes. Okay, so cholesterol synthesis is mainly like the excess acetyl-CoA, or from the diet, we convert glucose into pyruvate, then that intermediate cycle to convert them into acetyl-CoA. That acetyl-CoA, whatever based on our demand, most of it enter the citric acid cycle to release energy, but rest of them can be utilized to synthesize cholesterol, and we can store the cholesterol in our body. So when there is a need, it can be reutilized. Okay, so that is what is the significance of cholesterol synthesis. Cholesterol as such, the chemical composition is C27. H46O, that is 27 carbon atoms are present in the cholesterol molecule. Okay, the structure of cholesterol is very complex, so that much detail is not necessary. Cholesterol synthesis mainly occurs in liver, but also in intestine, adrenal cortex, etc. So within the cell, it occurs in cytosol and endoplasmic reticulum. That is what is ER. Okay, so the first step in cholesterol or input in cholesterol synthesis is acetyl-CoA. Okay, so here I have mentioned acetyl-CoA and acetoacetyl-CoA. So this acetoacetyl-CoA is formed by the combination of two acetyl-CoA molecules. So altogether, how many acetyl-CoA over here? How many acetyl-CoA molecules are in the input stage? Very good, 3. So one acetyl-CoA, then here acetoacetyl-CoA is 2 molecules of acetyl-CoA joining together with acetoacetyl-CoA synthase to form acetoacetyl-CoA. Okay, so acetyl-CoA plus acetoacetyl-CoA acted upon by enzyme HMG-CoA synthase to form HMG-CoA. This is a major energy compound. So HMG-CoA is beta-hydroxy beta-methyl glutyryl-CoA. It also is an intermediate compound in the metabolism of branched chain amino acids. So HMG-CoA is beta-hydroxy beta-methyl glutyryl-CoA. It is also an intermediate in the metabolism of branched chain amino acids. Okay, so this is the first step in cholesterol synthesis. Now, this HMG-CoA is acted upon by the HMG-CoA reductase enzyme and is converted into mevalonic acid or mevalonate. So this is a rate limiting step in cholesterol synthesis. Okay, so at this stage, the rate limiting enzyme is HMG-CoA reductase. Don't confuse, it is not synthase. In cholesterol synthesis, the rate limiting enzyme is HMG-CoA reductase. That is conversion of HMG-CoA to mevalonic acid or mevalonate. That is a rate limiting step. Now, this mevalonic acid is acted upon by decarboxylation enzyme to form isoprenoid units. So those are all complex structures with multiple carbon atoms. Okay, so isoprenoid units, almost around 6 isoprenoid units enter the reaction, or they are synthesized to make out ultimately get converted into cholesterol. Okay, so that inbuilt detail is not needed. This much is sufficient. Mevalonic acid is acted upon by a decarboxylation enzyme to form isoprenoid units. Next stage is synthesis of squalene by squalene synthase. So isoprenoid units gets converted or acted upon by squalene synthase to form squalene. So squalene is said to be a 30 carbon compound. Okay, and ultimately squalene. So in between squalene and cholesterol, around 20 complex reactions occur, and ultimately squalene gets converted into cholesterol. So mainly all those reactions are configurational changes. rather than removing an addition. They're just configurational changes happening. So ultimately, cholesterol formed will be a 27 carbon compound. So in between, and there are so many intermediates, and one of the intermediate which is significant is lanosterol. Okay, so ultimately squalene gets converted to lanosterol, and lanosterol is what gets converted into cholesterol at the final stage. So that much is sufficient in cholesterol synthesis. Remember, the late limiting step is HMG-CoA reductase. Okay, now we will have a mnemonic so that you can remember cholesterol synthesis. That is, all are happy mastering in some competition. So all A4 acetyl-CoA, again R A4 the acetoacetyl-CoA, H for HMG-CoA, mastering M for mevalonic acid or mevalonate, then I for isoprenoids, S for squalene, and competition C for cholesterol. So this is a mnemonic to remember the intermediates in cholesterol synthesis. And in cholesterol synthesis, at this rate limiting step, we utilize 2-NADPH to produce 2-NADP+. So actually, by cholesterol synthesis, we are utilizing energy in the synthesis of cholesterol. So 2-NADPH molecules are getting converted into 2-NADP+ compound. Okay, fine. So all these inputs lead to formation of one molecule of 27 carbon cholesterol. Okay. So you can just take a screenshot of this with the mnemonics. Done? Shall I proceed? Yeah. Now, we'll discuss a few questions based on this, and then we can move to the next cycles. The major lipid that make up the cell membrane. Yes, phospholipids, right? Triglycerides are the lipids that are the major forms that is getting stored in our body. Lipoproteins deals with the transportation of cholesterol and the triglycerides or triacylglycerol. Glycolipids are involved in the structural composition of various compounds, like the ganglions, the brain cells, and everything. The lipid which accumulates in fatty liver. It is triglycerides. Okay. So the lipid which is seen to accumulate in fatty liver is triglyceride or triacylglycerol and not cholesterol. Okay. So nowadays, apart from cholesterol, rather than cholesterol, triglycerides are getting more importance because they are supposed to be the greatest type of lipid getting stored in the body in various organs and causing a lot of issues. So triglycerides, we should have a check on them because that is the major type of lipid getting stored in the body rather than cholesterol. Okay. The essential fatty acid, which is present in breast milk, needed for development of brain and retina during neonatal period is. This has been asked for your NEET examination. It's a very important question. Its answer is B, docosahexaenoic acid. So this docosahexaenoic acid is formed from the essential fatty acid, that is linolenic acid. Okay, linolenic acid, when we take through diet, it gets converted into docosahexaenoic acid or DHA. Okay, and that is also present in human breast milk. So that is very important for the development of brain and retina for infants or during the neonatal period. Okay. So that deficiency can lead to various neural tube defects. That is why during pregnancy, we prefer giving tablets with folic acid as well as DHA because both are important to prevent neural tube defects and other complications. Rate limiting enzyme in cholesterol synthesis. Yes, it is HMG-CoA reductase, which convert HMG-CoA to mevalonate or mevalonic acid. Okay. So 2-NADPH is utilized in the step. Hypolipemic agents act on. So hypolipemic agents means, can you name a few hypolipemic drugs that we use? Or that group? Accurate, okay, you have a term, yes, statins, very good, statins, no? Atorvastatin, lovastatin, like that. So they're called as statins, right? So those are the hypolipemic agents. So their major role is they prevent the synthesis of cholesterol. So where should they act? If you have to inhibit cholesterol synthesis, where should they act? They should act on the rate limiting enzyme in cholesterol synthesis, that is HMG-CoA reductase. Okay. Which is the site for synthesis of cholesterol. So this is actually they're asking within the cell where cholesterol is synthesized. So the major site is liver, then comes intestine, adrenals, etc. But within the cell, they are synthesized in cytoplasm as well as endoplasmic reticulum. The starting molecule in cholesterol synthesis is, yes, it is acetyl-CoA. Don't confuse with option B. Acetyl-CoA and acyl-CoA, both are different. Okay. Which of the following statement is not true? Cholesterol is amphipathic in nature. What is meant by amphipathic? Amphipathic means it is both not catabolic, it is both hydrophilic as well as hydrophobic. That is what is called as, yes, both hydrophilic and hydrophobic. So cholesterol actually is amphipathic. It is both hydrophilic as well as hydrophobic. So statement A is true. Now, statement B, starvation inhibit cholesterol synthesis. Starvation, during starvation, will the energy, like, will there be cholesterol synthesis? Cholesterol, when you synthesize cholesterol, it is a storage form. So energy won't be released. We are utilizing energy in cholesterol synthesis. There is no production of energy. So in starvation, there is no cholesterol synthesis. So option B also is correct. It is a true statement. Now, what about option C? We have asked a false statement, right? So what about option C? T4, that is thyroid hormone, increases cholesterol synthesis. Okay. What happens in hyperthyroidism? Just you all know, no? Just it's not no need to think very complicated. When there is patient with hyperthyroidism, the patient will be very much lean because their metabolic activity will be, it is kind of a wasting disease for them, right? While hypothyroid patients will be obese. So in hyperthyroidism, that is whenever the T4 is increased, their metabolic rate is increased, body metabolic rate is increased. So as a result of this, they have told T4 increases cholesterol synthesis. That is wrong. The body energy is getting wasted out. So cholesterol synthesis is not happening. Instead, the cholesterol, whatever is stored in the body, is getting utilized in hyperthyroidism. The patient is becoming lean. Just think in a simple way. So that is option C is the right answer. Okay, because that is a false statement. When the patients with hyperthyroidism, since the basic metabolic rate is increased, even the stored deposits in the body also will be wasted out, releasing energy. Okay, so option C is the right answer. Energy needed in the rate limiting step of cholesterol synthesis. So that we have already told, it is we are utilizing NADPH. 2 molecules of NADPH is getting utilized in the rate limiting step of cholesterol.